We've talked about lipids, we've talked about their structure, we've talked about their roles. Now let's talk about breaking them down or build them up. So, lipids play unique roles in living organisms, largely due to their hydrophobic structure, because most everything else in an organism is hydrophilic. They are primarily responsible for the integrity of individual cells and the intracellular components. Now, what we're going to focus on is the synthesis and degradation of major classes of lipids. Acetyl-CoA, which we have talked about previously, is a central metabolite in lipid metabolism, either substrate or product for most lipid-related metabolic processes. Here is coenzyme A. Big old bulky molecule. We haven't really talked about all of it. We've kind of narrowed our scope in our discussion of it. It's got an acetyl group. There's kind of the acetyl-CoA that we've talked about previously, where we've got a carbon-sulfur bond and then CoA. Well, here's the CoA that follows. Let me make sure that here's the CoA. It's kind of nice to just talk about it as CoA. So our summary of metabolism is basically displayed right here. What we've focused on so far is we've focused on carbohydrates, getting the citric acid cycle, going to the electron transport chain, and gone. Acetyl-CoA is a key factor in all of that. And if you look kind of outside of that range, well, acetyl-CoA is also going to play a role in protein and amino acid metabolism, going to acetyl-CoA, just as fatty acids go towards acetyl-CoA. So acetyl-CoA is kind of a nice little central hub. Now, fatty acids are important and an efficient source of energy for many living cells. We talked about previously how they're very efficient because they can pack so well. The average U.S. diet, 30 to 40% of the calories that the average American intakes are provided by fat. Triacylglycerols are digested in the small intestine. They form glycerol and fatty acids that are absorbed. Newly formed triacylglycerols are packaged with other lipids in the form of chylomicrons and transported to the blood. Adipose tissue, or adipose cells, skeletal, and cardiac muscle convert triacylglycerol into glycerol and fatty acids. Those fatty acids are taken up by cells. Glycerol is transported to the liver. Now, processing of dietary fat. Depending on an animal's metabolic needs, fatty acids may be converted to triacylglycerols through a process known as lipogenesis, when energy levels and metabolic fuel is high. Insulin is going to activate this synthesis. They also can be converted to provide energy through the process known as lipolysis, when our energy and glucose levels are low. Glucagon is going to stimulate this lipolysis, which is also known as beta oxidation. In this case, fat is going to be mobilized. This can also make phospholipids when the cell needs to make membranes and make them for the purpose of providing a cell structure. Now, much of lipid metabolism requires activation of fatty acids. So the way that a fatty acid is going to be activated is, well, here is a free fatty acid. Here's our R group indicating that aliphatic chain. Our carboxylic acid is right here. Then we have an ATP molecule and coenzyme A. What we're going to do in order to do this is use the enzyme acyl-CoA synthase. And that's going to produce, well, it's going to cleave ATP into AMP and release inorganic pyrophosphate. So what we have to have is acyl-CoA synthase and inorganic pyrophosphatase. Ultimately, this is going to be activated by a fatty acid product called acyl-CoA. Our acyl-CoA molecules can then either be used for lipogenesis or fatty acid degradation. Lipogenesis, step 1. Most triacylglycerols are synthesized in the liver, stored in adipose tissue. Okay, so our triacylglycerols, or our TAG, are going to be synthesized in the liver and stored in adipose tissue. Backbone for the molecule used to form triacylglycerol can be a glycerol 3-phosphate. Glycerol is converted to glycerol 3-phosphate using 1 ATP. Our first acyl group is transferred to glycerol 3-phosphate to form lysophosphatidic acid, or lysophosphatidic acid. This is going to occur in the liver. Next, it can also be from dihydroxyacetone phosphate. Now, these two things are very, very similar to intermediates in glycolysis. Now, DHAP, glucose is converted into DHAP through glycolysis. This is going to occur in adipose tissue. DHAP is going to be acylated, then reduced by 1 NADH to form lysophosphatidic acid. So when you think about lipogenesis, I want you to think about a goal of getting to this molecule of lysophosphatidic acid. Now, there's a lot to process with respect to lipogenesis, but it's somewhat straightforward. Let's start out with two molecules. The first molecule is glycerol, and then the next molecule is dihydroxyacetone phosphate, which was derived from glucose. Now, the first one that we're going to tackle is this glycerol. Glycerol undergoes some pretty quick processing to get to a point where it's lysophosphatidic acid, which is I'm going to go ahead and highlight that as kind of our goal and our objective. Okay, so glycerol is phosphorylated by glycerol kinase to make glycerol 3-phosphate. Then glycerol 3-phosphate is acylated to form lysophosphatidic acid. So glycerol's done. Glycerol has formed lysophosphatidic acid with the help of two enzymes. Glycerol kinase phosphorylates it. Then we have an acyltransferase reaction with the help of coenzyme A. Glycerol 3-phosphate acyltransferase makes lysophosphatidic acid. Dihydroxyacetone phosphate, on the other hand, has some different routes that it can take. Here is our structure for dihydroxyacetone phosphate. One route that it can take is it can go through glycerol 3-phosphate dehydrogenase and form glycerol 3-phosphate. Then it follows the exact same pathway as the glycerol that we just phosphorylated. It gets acyltransferase, or it undergoes an acyltransferase reaction, and we result with lysophosphatidic acid. Dihydroxyacetone phosphate can also undergo an acyltransferase reaction immediately with the enzyme dihydroxyacetone phosphate acyltransferase, so DHAP acyltransferase, which gives us acyl dihydroxyacetone phosphate. So we've got our acylated dihydroxyacetone, and now what we need to do is generate some NADP from our NADPH. So we've got our acylated and phosphorylated, our lysophosphatidic acid, but what we need to do is get rid of this carbonyl oxygen right there. And so we're going to use acyl dihydroxyacetone phosphate reductase. And there we go, lysophosphatidic acid. So the way that I want you to think about this is step one, dihydroxyacetone phosphate or glycerol. Your aim is to get to lysophosphatidic acid. Once you've done that, you can move on to step two. The second step is, and keep in mind, we're trying to produce a triacylglycerol. So we've added one acyl group. Now we've got a phosphate group on there. Now what we need to do is use another acyltransferase. So we're going to add another, we're going to form another ester linkage right here and add another acyl group. So now what we've produced is phosphatidic acid. So we've got our phosphate group on carbon number 3. We've got an acyl group on carbon 1 and carbon 2. So we're in great shape there. This is awesome. So after 2 steps, we've got 2 of the acyl groups added. Now, one route is that phosphatidic acid, well, it still has that phosphate. So we could send that through phospholipid synthesis and make some modifications to that phosphate group, or we could just continue processing it. And we're going to continue processing it, storing those fatty acids. And what we're going to do is we're going to take phosphatidic acid right here, and we are going to convert it to diacylglycerol by removing our phosphate from carbon number 3. The result is a diacylglycerol molecule. We've got 2 fatty acids there, and then we have a hydroxide group on carbon number 3. We're going to use the enzyme diacylglycerol acyltransferase to add our third and final acyl group on there. So There we go. We've made a triacylglycerol molecule. There are other routes that we could take this. As we saw previously, we could have gone to phospholipid synthesis, or we could have made a monoacylglycerol by removing one of our acyl groups. But that diacylglycerol is going to be activated to form our triacylglycerol with the help of an acyl transferase. So we've got lots of acyl transferases, and in the process, we've utilized a good amount of acetyl-CoA, or sorry, coenzyme A. So this is the whole thing that we just talked about. The way that I want you to think about it is I want you to think about the bottlenecks. What are the key points? Glycerol, dihydroxyacetone phosphate, go to lysophosphatidic acid. Then we go to phosphatidic acid, diacylglycerol, triacylglycerol. Essentially what we're doing, we've got to start out with something that's phosphorylated and then add some acyl groups. And we do that with acyl transferases. So if you wanted to distill it down and make it as simple as possible, maybe the most important enzyme through this process is an acyl transferase. All right, so that is going to be where we stop talking about triacylglycerol synthesis, and now we're going to pivot and we're going to start talking about triacylglycerol degradation. This is referred to as lipolysis, and this is going to generate a huge amount of energy. This occurs during fasting, vigorous exercise, and in response to stress. Now, a cascade mechanism releases fatty acids and glycerol from triacylglycerol, and this is going to be the case whenever glucose levels are super low. Glucagon and epinephrine are released. These hormones bind to plasma membrane protein receptors, elevating cytosolic cyclic AMP. Cyclic AMP acts as a secondary messenger, activating triacylglycerol lipase. That name lipase is similar to a phosphatase. What it's going to do is it's going to cleave off a lipid. It's going to cut a lipid, much like a phosphatase cuts a phosphate off. So here's that signaling cascade where we have the activation of cyclic AMP, our secondary messenger, which is going to activate a protein kinase, which is going to activate triacylglycerol lipase. All right, we will continue on in the next video.